speaker this morning is Assistant Professor at the Samar State University. She had been in the service for 20 years and she is a licensed teacher. She finished her Doctor of Philosophy, major in Science Education at the University of Perpetual Health System Delta. Our speaker is designated as the Natural Science Chairperson, Chemistry Lab in Charge, and Extension Coordinator of the College of Arts and Sciences at the Samar State University. At the same time, she is the Laboratory Manager at the Analytical and Biochemical Laboratory of the Research and Innovation Center of the same university. Our speaker is also an active member of several professional organizations. She is a member of the Philippine Organization of Science and Technology Educators and also a member of the Philippine Association of Research Managers Incorporated, State Universities and Colleges Teachers Educators Association. Ladies and gentlemen, let us all welcome our speaker this morning, Professor Edelyn Echapare.
Hello everyone, I am Edelyn Echapare. I will going to share with you about a certain plant that can be found in Kaigangan in Samar. This plant is formerly used or utilized by the people merely for ornamentals, but through scientific exploration, this plant has become economically important. To start with, shown in the picture is a plant taken from Kaigangan in Samar and were submitted to National Museum Botany section for its proper identification and authenticity. And it has been found out to be Cristela dentata, which is a pteridophyte that belongs to the family Thelipteridaceae. It is a soft fern with long green fronds. Another fern plant that can be found in Kaigangan in Samar that belongs to the same family with that of Cristela dentata is Macrothelipteris. This particular fern plant is a perennial fern that grows in disturbed areas including roadsides and forest edges and behaves as a weed. It spreads via spores in wind and water, but can also be spread via stolons and rhizomes in garden waste. The Thelipteridaceae are abundant species in summer, but being designated because of its low livelihood potential and economic use. So we get interested to these plants. From this, we started scientific exploration on some fern plants. We were able to analyze the phytochemical content of the plant and also examine its antibacterial properties. We collected samples from Kaigangan and brought these samples to SSU laboratory for antibacterial evaluation and phytochemical screening. For the phytochemical screening and extraction, a particular solvent has been utilized in the extraction process. A specific solvent, for example, if the test is for esterols and terpenes, alcohol is used as solvent in the extraction. For the antibacterial properties evaluation of the plant sample, we use the disc diffusion method in which the zone of inhibition is the reference how susceptible a particular extract to bacterial growth. And for the results of the conducted experimentation, the plant extract contains saponins, tannins, organic acids, and alkaloids. It was also proven that the plant extract has an ability to prevent bacterial growth, either gram-positive or gram-negative bacteria. Statistically, the antibacterial activity of the plant extract has no significant difference with that of either gram positive or gram negative and with that of the positive control. So this result entails that the plant extract has the potential to prevent bacterial growth. From the result of the study we conducted, we come up with a product. So here comes the fern soap. The fern soap is a germicidal with the extract additive, which is the fern extract, and the product has a moisturizing effect, and it's all organic. How this soap was being made? Watch this video. Physicochemical assessment was conducted with the formulated soap. One, for instance, is the weight loss on curing. So the samples were allowed to cure at room temperature for 4 weeks during the time of the weight of the samples were taken after every week interval. So the weight loss was determined by subtracting the initial weight with that of the weight of the sample every 7th day of each week. Another physicochemical parameters that has been evaluated were the moisture content, foam height, and foam retention. Moisture content was measured using the thermogravimetric method. With this method, the moisture is determined from the weight loss of the sample dried by heating, in which a 5 gram of each sample was run in a moisture analyzer balance was set at 160 degrees Celsius and the rate of moisture changing to 0.05%. For the foam height, the 1 gram of the soap sample was dissolved completely in 100 ml distilled water, and then take the 10 ml of the solution into the separate boiling tube. Then close the tube with a rubber cap and shake the tube vigorously for 3 minutes and let the solution stand for 10 minutes and measure the foam height. So we recorded the observation for 3 consecutive experiments, and the mean was able was computed. And then for the foam retention, time refers to the time for which the foam produced by the soap retains. So it has been, the above procedure has been repeated and the foam interval was measured for about 5 to 10 minutes. Other attributes of the soap were also evaluated using the sensorial evaluation. We made a questionnaire. We utilized The sensory 9 hedonic scale evaluation for gathering participants' preference as to fragrance, foaming, rinsing, moisture, irritancy, and overall acceptability of the soap.